Welcome to today's instructional video where we'll be discussing some fascinating methods in gene transfer technology. Our focus will be on the gene gun and alternative techniques for delivering genetic material into cells. I'm Daniel, and with me today are two experts in this field. Genesis, who will talk about the gene gun, and James, who will discuss alternative techniques. Let's dive right in.
Thanks, Daniel. The gene gun, also known as the holistic particle delivery system, is a method where DNA is literally shot into target cells. This technique is especially useful for delivering DNA into plants, animal tissues, and even cultured cells. The way it works is quite interesting. Microscopic particles, often gold or tungsten, are coated with DNA and then propelled into cell using a burst of helium gas. This process allows the DNA to penetrate the cell wall and integrating with the cell's genome.
So basically, you take those little cartridges that you have a picture of over there. You slip them in here. You can hold 12 of them in there.
Okay.
Okay, that goes into the gun here,
Wow.
like that.
It's like
It's a
a
12-shot revolver.
Yeah.
Yes. Okay. Then what you do is you plug this into the outlet, and what that does is that's what the trigger operates on. So pushing the trigger will release a stream of helium, which is attached to this gun. And basically, what happens is when the helium is released, it goes through those little cartridges, rips the gold beads off of the cartridge, and they go flying down the end of the barrel of the gun, and they hit whatever you have the end of the gun pressed up against. So this is like an animal belly or the side of an animal, or you can do tissue culture, cells, whatever you want. You can do plants.
And what are these carrying into the body? What's the goodness of these?
Okay, well, basically, the little gold particles have, in our case, DNA vaccines attached onto them, or precipitated, stuck onto them. And basically, once the gold bead gets into your nucleus, the DNA vaccine on there will dissolve off the bead. And since it's
One of the main advantages of the gene gun is that it doesn't rely on a biological carrier like a virus, which can reduce the risk of immune responses or other unwanted effects. It also allows for precise targeting of tissues and is particularly effective with a hard-to-transform species. However, the gene gun has its downsides. It's a bit random in terms of where the genetic materials lands, which can lead to unintended mutations. There are also the cost factors, as the setup can be expensive.
Thanks, Genesis. While the gene gun is a powerful tool, it's not the only method available for gene transfer. One of the most common alternatives is agrobacterium-mediated transformation. This method used in Agrobacterium chumsface, to deliver DNA into plants. The bacterium naturally infect plants and can insert genetic materials into their genome. Scientists have harnessed this capability for genetic engineering, making it highly efficient in widely used methods. Another widely known technique is CRISPR-Cas9, which has taken the world by storm in recent years. CRISPR doesn't just insert genes, but can edit them directly, allowing for precision gene editing. This method can be applied to a wide variety of organisms, from plants to animals and even humans. It's much more precise than the gene gun in terms of where the genetic material is inserted.
The CRISPR-Cas9 system represents genome editing technology that has revolutionized molecular biology due to its precise and site-specific gene editing capabilities, which essentially allow for an unprecedented level of control in manipulating the genetic information of a living organism. How does this work mechanistically, and what are its applications? Let's get a closer look now, starting with some historical context. In 1987, Atsuo Nakata and his team of researchers from the Osaka University in Japan first reported the presence of clustered, regularly interspaced, short palindromic repeats, abbreviated as CRISPR, in the Escherichia coli genome. These refer to short, repeated sequences of DNA nucleotides found within the genome of prokaryotes. These sequences are the same when read from 5' to 3' on one strand of DNA and from 5' to 3' on the complementary strand, and are therefore described as palindromic repeats, just the way that we refer to words like race car or kayak as being palindromes, because they are the same whether read forwards or backwards. This was further reported in both gram-positive and gram-negative bacteria, along with archaea, leading to the obvious question regarding the relevance of CRISPR to these organisms, which drove research for some time. Later on, in the mid-2000s, the functionality and importance of CRISPR was first realized in prokaryotes. As it turns out, the CRISPR system is a key component of their adaptive immunity, which protects these prokaryotes from attack by viral DNA, bacteriophages, and plasmids. That's right, it may seem incredible, but even unicellular bacteria have a very basic immune system. Recall from our studies in the immunology series that adaptive immunity refers to the immunity that an organism acquires after exposure to an antigen, either from a pathogen or vaccination. Vaccination, for example, results in a form of adaptive immunity in humans, since the body is exposed to antigens and forms antibodies in response, which contribute to the development of the immunity.
These are both fascinating methods. To recap, the gene gun is made for hard-to-transform species and doesn't use biological carriers, while agrobacterium-mediated transformation and CRISPR Cas9 offer more precision. But there must be some pros and cons consider when choosing these techniques. James, what are the pros and cons of using these alternatives? Sure. For agrobacterium-mediated transformation, the main advantage is the efficiency, especially in plants. It can precisely insert gene with fewer off-target effects. But it's limited to plants under natural susceptible to agrobacterium, and it's not as fast as the gene gun. With CRISPR Cas9, the precision is unmatched, but it can be quite complex, requiring expertise to design the guide RNAs and ensure no off-targets editing occur. There are also ethical concerns with use CRISPR in animals and humans, given its power to make permanent changes in DNA. Thanks for that comparison. So, in summary, the gene gun offers a fast and physical way to introduce genetic material, whereas other methods like agrobacterium and CRISPR offer more precision but come with their own sets of challenges. Depending on the project, scientists will choose the method that best fits their goals. Thank you, Genesis and James, for sharing your expertise today. And thank you for watching. We hope this video helped clarify the different gene transfer techniques available. If you have any questions, feel free to drop them in the comments. See you next time.